Okay, I hope we are online. Good afternoon. I am Guillermina Lopez Bendito from the Institute of Neuroscience in Alicante. And it's my pleasure to be joining this Neuromatch meeting for the first time for me. And also it's a real pleasure to have Simon Hippenmeyer as a speaker today. And Simon and I met a few years ago, and we are good colleagues and so on. And so Simon, since I think 2019, 1 year ago, something, got the full professor position at the IST Austria. But before that, he was also in the IST for a few years as an assistant professor. And Simon is a very well-known worldwide as I would say one of the fathers of this technique, MADM technique for labeling lineage tracing of neurons and cells in the brain. And this is a technique that he used widely and to show how the cell diversity occurs in the cortex and other brain regions, and in which he had been using for a long time. So just a little bit of a background for the ones that don't know Simon. So Simon did the PhD with Sylvia Harber, and then he moved to Stanford to do a postdoc with Li Chun Lu. In several years, I think 5 or 6 years he was there, 6 years maybe. And then he went back, as I said, to IST Austria, and he got also one of these prestigious ERC grants, and he had been working in very nice projects, so on, regarding the molecular mechanisms for neural stem cell production and lineage progression. Also, he has been working in genomic imprinting that maybe he will be telling us today in the cerebral cortex. And I think he has also future plans for looking at lineage progression during evolution, I think. So thank you, Simon, for accepting the invitation to talk in the Neuromatch. And thank you, everybody, for connecting. And I hope you will enjoy Simon's talk. So thank you.
Okay, thank you, Guillermina. I'm going to share my screen. Okay, I hope you can see this now. I'm not seeing anyone anymore, but thank you very much, Guillermina, and also all the organizers for this wonderful initiative, this Neuromatch for us developmental biologists or neuroscientists is actually, I think, the first time happening. But I really think in these days, this is a wonderful initiative. So thank you for the invitation also to speak here. Okay, so the main question, or one of the bigger questions we're addressing currently in the lab, is really how to produce a brain with correct size and how to generate all the different cell types that you find there. And we currently focus on the cerebral cortex, and in particular, we are using mass genetics to address this question. So already a long time ago, Ramani Kahal, of course, noticed that not all the cells in the cerebral cortex are the same. And today we know that there's actually specific factors or transcription factors or even transcriptome signatures that define the different cell types. This is now an image from the mouse. So you can see the laminated organization, which is actually, yes, evolutionarily conserved of these different cell types. And importantly, the laminar position dictates the wiring of these cells. And we do know if something goes wrong in the production of not the right number or the different cell types, basically due to genetic mutation, that this can lead to neurodevelopmental disorders such as schizophrenia and autism. Or in much more extreme cases, it can really be the mutation of a single gene that leads to a drastic disease, microcephaly with a smaller brain compared to normal, or lissencephaly with complete disorganized laminar structure, and the folds are also very much not there. So there is a huge sensitivity in the process to produce all the right cell types and the right amount of cells. Now, while in most of these diseases, the etiology is actually not so clear, the nature of genetic mutations is becoming clear increasingly. And we believe that this can give us maybe some insights, but also may imply some general principles. Now, in our lab, we take this as an inspiration and are really interested in how neurostem cells during development are able to produce all the different cell types here in the distinct layers in the cerebral cortex. And as Guillermina also already mentioned, we're using mainly quantitative lineage tracing and genetic manipulation at single cell level in mice. And the prime methodology that we are using is called Mosaic analysis with double markers. Again, this is a genetic method in a mouse. And importantly, what MADM, this technology can really do is it can capture progenitor stem cell division patterns at single cell level. And this is really a method that I got inspired when I was in Stanford, a postdoc, and that was originally developed by Huisson in the laboratory of Litchfield Low. Now, here is really the principle how MADM can work. So you're starting with a cell that is not labeled, but then you induce this MADM event. I will not go into technical details. I mean, it's published quite a while and there's literature you can read. But the important thing is that the two sister cells express two different markers, one's red, one's green. And depending on the further proliferation pattern of these sister lineages, either more symmetrically or asymmetrically, you can really infer the cell division patterns with high accuracy. Now, on top of it, we also can introduce genetic mutations of interest, specifically in one of the lineages, here in the green lineage, and then compare the effect at single cell level of that mutation when compared to wild type. And for many years, actually, we're working towards a resource that we now completed. It's actually here in bioRxiv by Contreras et al. That allows more than 96% of all mouse genes to basically subject to such an assay. Now I show you one clone here at higher resolution, and this is really what it is. So basically, all the cells that are labeled here is the result on the output of a single stem cell, which we induced at this MADM event at embryonic day E11 and analyzed at 3 weeks postnatally. You can appreciate the quantitative nature of the approach. You can go and count the different labeled cells here in the different layers. There's also Glia cells, not only projection neurons. And over the last years, we really have analyzed hundreds of these clones with the aim to see whether we can get to a general framework of lineage progression in cerebral cortex. And this is the result, the summary, basically, what we came up with. So we have now an initial phase of neural stem cell division patterns. So this is symmetric divisions before the radial glia progenitors then start to produce neurons of all the different layers by asymmetric division. Now, the intriguing part about this finding, at least to me, is that once these radial glia cells start producing neurons, we can predict the overall output, which is about 8 to 9 neurons in total, that then assemble here or distribute over these different layers. Now, not only that, also about 1 in 6 can be predicted to retain the potential to produce glial cells, such as astrocytes and oligodendrocytes. Now, while there's many unknowns, of course, in the framework here, we are now really interested in the molecular and cellular mechanisms that drive the lineage progression, or the switch from symmetric to asymmetric, the sequential production of neurons, and then the switch to glia production. And today, actually, I will tell you 2 brand new stories. So unfortunately not imprinting as Guillermina was maybe expecting, I'm sorry, but I really want to present recent data that we got in the lab. But basically, the inspiration for the first project I'm going to tell you is really that the laboratory of Denis Chabaudon in Geneva recently found in a very beautiful paper here, published about a year ago, is that there's a temporal patterning of radial glia progenitor cells as they produce the neurons. So in other words, they isolated with single cell resolution the transcriptomes at every step of neuron production here, and they could identify intriguing fingerprints that seem to pattern the stem cells when they produce the neurons. Now, we have then carried out a candidate gene approach where we looked at EED, which is part of the PRC2 complex, and found actually that lineage progression in such a mutant is affected. In other words, you anticipate way too fast the neuron production, and this results actually in limited output of neurons and a severe microcephaly. Now, before I go into detail, I want to tell you a little bit more about this PRC2 or polycomp repressive complex 2. It's actually regulating chromatin, and it's required for cortical development. So this was known before, but what is PRC2 complex doing? It's actually methylating histones, and that leads to gene silencing. So in other words, you're repressing gene expression. Now, if you're losing part of the complex, namely this EED, for instance, as we did together with Denis, you're actually losing also methylation, and you have no gene silencing, and you have increased accessibility and expression of target genes. And thereby, this correlates with a very severe microcephaly, as you can notice here. So we then measured cell cycle, and indeed, so there is an increased cell cycle exit during especially upper layer neuron production. There's also a thinning of the ventricular zone, so there's less stem cells over time, and as you can see, this results in a severe microcephaly. So now we were really wondering what is going on and what are the mechanisms of the loss of function here. And we're addressing three questions now in this part of the presentation, namely, what are the mechanisms of this PRC2 complex in temporal lineage progression? And also, to which extent is that function cell autonomous or necessary at the global tissue-wide level? And we have seen that there's a problem with neuron production if you're losing that complex, but there may be other functions in lineage progression, which we're also looking at. Now, this is the project really driven by a very talented postdoc, Nicole Amberg, together with Florian Pauler, who is a bioinformatics expert. Now, the paradigm that Nicole was using is actually really to try to address, firstly, that question, namely, how we can dissect intrinsic gene function and the global or stem cell niche tissue-wide mechanisms due to loss of function at single cell level. And for this, we're having two paradigms here. In the first one, we're knocking out a candidate gene. Now, in this case, it's going to be EED from the PRC2 complex, just in single cells with a normal environment. And we can trace the phenotypes of these mutant cells with high single cell resolution and thereby infer a cell autonomous gene function. Now, as you know, usually we can also induce a global knockout or a conditional knockout. And also together with this labeling, the MADM labeling, you can see that these mutant cells are homozygous mutant for that gene, but it's the same like these cells. And any difference in the phenotype of the green cells in such a paradigm where we have a mutant background in comparison to this paradigm where we have a normal background actually can provide some hints at these non-cell autonomous effects or mechanisms. So this is what Nicole then did, and here is the summary of the data. So we have a control here with MADM labeling. It's a bit low res, but here it's at higher resolution, so all the cells are wild type. Then we have the conditional knockout. You already have seen we have a dramatic microcephaly here, which can reach up to 50% of the normal cortex size. And then we also induced the genetic mosaic with sparse deletion of the gene in single cells here with a normal environment. And we didn't see any microcephaly here, but even more surprising was when we counted mutant cells and compared them to wild type cells, we saw a ratio of 1:1. So in other words, there were no less green cells than red cells. And also that shows really that cell autonomous loss here of EED does not affect neurogenesis as such, at least not to the level that we have when we mutate the gene everywhere. So this is really providing evidence that EED itself or the PRC2 complex is not required cell autonomously for cortical neurogenesis. Now, to address this a bit more in detail, we went back to single clonal analysis, so where we really induce single stem cells. And you can see that if we do this with a conditional induction of this MADM clones at embryonic day E11, look 2 days later, we basically don't see a difference between the number of EED mutant cells and wild type cells here at this level. We also look a little later at E16 at the end of neurogenesis. Also here, when we compare mutant cells to wild type cells, it's roughly the same, so there's no significant difference. So that really shows that at a single radioglia progenitor level, introduction of a mutant lineage here compared to the wild type lineage doesn't affect any of the output of these cells. Now, this really shows that there's no cell autonomous function, but then we still measured the total output. I told you earlier, there's a unit of about 8 to 9 neurons, and which you can really count here when you induce the tamoxifen and induce the MADM event at about embryonic day 12, when neuron production starts roughly, and then look at 3 weeks of age. Now, also in this case, the total unit size here is not changed. So black is EED mutant and wild type is the same. It's about 8 to 9 neurons. And also in superficial or deeper layers, there's no difference in the total output within single clones here. So that really seems to confirm that cell autonomously PLC2 is not required for cortical neurogenesis, and that the dramatic microcephaly we see in the conditional knockout is rather the effect of a systemic loss of that gene. Now, this is summarized here. So basically, having single cell knockout, you have normal neurogenesis. With a conditional knockout, all cells mutant, you have a major defect in neurogenesis and microcephaly. And so we wanted to know what's different actually in these two paradigms. What's different in the green cells when all the cells lack that gene, or in the green cells with a normal environment. To this, we then isolated tissue, dissociated the cells using FACS sorting, and then extracted mRNA and did RNA sequencing to get a transcriptomic fingerprint of those cells and compared to control, and then again the mutant cells in the mosaic or mutant cells in the conditional knockout. Now, as a control first, we measured EED expression. So in all our paradigms, regardless if it's sparse or conditionally, we actually have downregulation or ablation of these EED transcripts, whereas in the wild type or in the control, there's a robust expression level at the times that we measured. Now, when we look at deregulated gene expression, it's actually dramatic. So now when we compare the green cells from here with the green cells from there, we saw that at about embryonic day 13, which seems to actually mark the onset of the problems in neurogenesis in the conditional knockout, we start seeing deregulated gene expression. And that is quite dramatic if you look in the time course. So there's more than thousands of genes that are upregulated, which in principle makes sense because it's a repressor. So you're losing repression, you have ectopic upregulation, but we also have downregulated genes, and these likely are secondary effects that may also play a role. Now, if we look at pathway enrichment, there's all the famous pathways that seem to be affected, the Wnt signaling, which we know is very important for neurogenesis, there's P53 signaling pathway, cell cycle, etc. If we look a bit more in detail, especially at E13.5, when the microcephaly seems to start, we see there's lots of deregulated genes, but most importantly, if we measure cell cycle dynamics or EDU incorporation into the MADM cells, we see specifically in our CKO, where all the cells lack EED, there's a sharp drop, but not in the mosaic. Again, this shows that cell autonomously, cell cycle is not affected if you're losing EED, but then systems-wise, it is affected. The same is true for cell death or apoptosis-related pathways and genes. We see in our CKO, there's quite some upregulation of the apoptosis. This is also measured here, and this is not the case in the single cell or sparse knockout of EED. So this shows really that there's a dramatic difference in cell cycle and apoptosis, but only if you're losing that gene in all the cells, but not in sparsely the single cells. Now, microcephaly is often associated with mutations in these genes, regulating cell cycle and DNA repair and chromatin organization, as I just told you. But then in mice, what happens sometimes is that if people do loss of P53 experiments, that can rescue many of these microcephaly phenotypes. And so we ask whether our microcephaly phenotype can be also rescued by loss of P53. Now here's the data, and this is not the case actually. So I'll walk you through. So basically this is a wild type scenario. We have a regular cortex, a certain thickness. If we're losing p53, not much happens actually. So the cortex is more or less intact. Although we are not having dying cells so much anymore, so we measured that, of course. But then if we compare to, oops, what happened now? Sorry. If we compare to the EEG CKO, we have this massive microcephaly here, as you can see. This is also measured here, but then also losing p53 homozygously doesn't do anything, and the microcephaly is similarly severe. So that really shows that whatever pathways are affected, it's not acting through p53. Now we were interested to find out, okay, what target genes are we actually talking about that we now having ectopic expression? And to this, we went back to a study from the Huttner lab where they actually measured by ChIP-seq all the histone modification tags here. And so the rationale was, if you have this histone methylation, you wouldn't have gene expression at all. And if we combine this with our POC context, if you have deregulated genes where you don't have a tag, this would be secondary effects. And if you have normally histone methylation tag and you would have ectopic expression, this would be the POC to direct targets. So this is shown here. If you're losing the activity, you would lose the methylation. You're having ectopic high gene expressions. We have already seen this. There's many genes. And now combining this with the Huttner data set, we really can see that downregulated or upregulated genes where normally there's no histone modification, this would be secondary effects. And if there were a histone modification close to this gene, this would be actually the primary targets. And this is the data, so it's actually quite dramatic. So if you focus on the direct target, they are massively, there's a huge number of direct targets, about 180, and very little in the mosaic. But these seem to really repress genes for basic biological developmental processes. That was to be expected. But then more surprisingly was that the secondary effects are also much, much bigger in our conditional knockout. So there's 370 at only embryonic day 13.5. And these actually seem to really affect cell cycle, DNA replication, and these processes critical for neurogenesis. Now, when we do network analysis, we can connect direct targets to this cloud of secondary effects. And in fact, it's very specific for the conditional knockout. So for instance, this gene CDKN2A is only upregulated in the conditional knockout, but not in the mosaic and not in the wild type. So we believe that in this cloud of deregulated genes from a small set of direct target genes, we are affecting major pathways that control neurogenesis. So this really shows that there is an important role of EED PSC2 complex in lineage progression here, and the deregulation of a relatively small number of direct targets leads then to massive secondary effects, and those actually include cell cycle and apoptosis deregulated genes, but there's no rescue if we also lose P53. Now, this shows a really dramatic function of PSC2 activity in lineage progression and neurogenesis as such, but there has been in the literature evidence or suggestions that it may also affect production of astrocytes. However, the evidence is conflicting. Some say it does, others say it wouldn't, and so we took this as an opportunity to analyze astrocyte production in the absence of PSC2 complex. The data is shown here. So we have a control MADM, all cells are wild type, where we have about 20 astrocytes per square centimeter. Then if we're losing EED in the green cells, we have a dramatic reduction of astrocytes here, as you can also see from the overview. So that shows that EOC seems to be essential for astrocyte production. We also looked in a time course, and there's no rescue later on during development, so it's not a delay. So it's really, while a certain level of astrocytes are produced, this level is constantly low. So there seems to be an astrocyte production problem. We also measured at the clonal level using, again, our clonal induction paradigm. Also here we see there's a significant drop in astrocyte numbers per clone when you're lacking EED. Now, we also looked at morphology of these cortical astrocytes, and it's actually quite dramatic. So if you look in a time course, these, especially at 3 weeks of age, it's quite significant. So these astrocytes lacking EED are much smaller, and also the branching pattern is a very different one. So we wanted to also see a bit further with transcriptome analysis what goes wrong here. So we isolated astrocytes, again using a FACS paradigm, and then measured again gene expression differences of wild type versus mutant. And also here we see quite a dramatic deregulation of genes. There's not that many as in cortical neurogenesis, as you can see, but there is a significant amount of genes up and down regulated. What we think is particularly interesting is that actually genes related to synaptic signaling are ectopically upregulated here in the mutant cells, and that may be interesting in regard to the morphological phenotype as well. So this is something that I think we should look at further. Now, let me summarize this part here of the presentation. So I showed you that we have a global tissue-wide function for EAD or PSE2 for neurogenesis, and I showed you that actually it's a secondary effects due to deregulated gene expression which seem to be responsible for that phenotype. It's p53 independent on top of it, and cell autonomously, PSE2 seems to have no function for neuron production. In this slides before, I showed you that also once Glia cells are being produced, there's a cell autonomous function for astrocyte production and maturation. So there's a different function very clearly that is also required. I didn't show you the data, but there is also a function in adult neurogenesis, which we don't understand completely. So we are still looking at this. Now, I want to really focus also on the second part, which is way shorter, so it's don't be afraid. So it's not as long as this one, but it's an intriguing new finding actually that relates to asymmetric cell division. And as you can see here, during lineage progression of these radial glia progenitors, by asymmetric cell division, they produce neurons and or later astrocytes. And the field is actually quite mature in that sense. There's ways how to generate asymmetry in a neurostem cell division. So either you have the secreted signaling molecules, which may pattern the plane of division. Alternatively, also the whole tissue may be patterned and with some polarity cues that may instruct asymmetric cell division. Or there's also intrinsic components which actually drive the fate of the two daughter cells. Now, for the last few minutes, I really want to focus on an alternative theory, actually, which is the selective chromatid segregation model. So what do I mean with this? During cell cycle, we know that chromosomes or DNA is replicated, and there is always two sister chromatids. Now, this selective chromatid segregation model actually proposed that the two sister chromatids, although identical in DNA sequence during replication, they would acquire some kind of tag. So the hypothesis is that it's also epigenetic to distinguish themselves, and even further that this selective segregation of these two sisters to either the stem cell or the postmitotic cell would actually instruct the fate of these cells. So in other words, because there's epigenetic tags potentially, they may produce these non-equivalent sister cells that then would correlate with the fate of the daughter cells produced. Now, this sounds a bit spooky, but there is actually real evidence that this may really happen. In particular, in Drosophila, it was found in vivo that this non-random sister chromatid segregation happens during germ cell division, but also from cultured mouse embryonic stem cells in vitro. And what people have used to actually measure the segregation of sister chromatids is an assay involving interchromosomal recombination that can mark the two sister chromatids selectively and thereby enable the tracing of chromatid segregation in stem cell division. So again, we would have a cell in G1, then DNA synthesis happens, so you duplicate your genetic material, but then this X marks actually a recombination event. This happens, this can be actually induced artificially, but it also happens naturally every now and then. But that would produce these so-called recombinant chromosomes. So you can see the red and blue split as opposed to the parental blue and red chromosomes here. Now, depending how these recombinant chromosomes that can be traced now segregate, it can be that they go to the same cell and the non-recombinant one go to the other one. This is called set segregation, and again, this happens also in Drosophila and other organisms. However, if these recombinant chromosomes go to different cells, actually this leads to a loss of heterozygosity, especially in these distal parts. You see both parts here are red and here it's blue. Now, in principle, if chromatid segregation is completely random, you would expect a 50% segregation in that direction and a 50% segregation in that direction. However, as I already mentioned, if people use this in the embryonic stem cells, mouse embryonic stem cells, and trace chromosome 7, for instance, what they found is in neuroectodermal stem cell lineages, there's actually no G2X, so no segregation in that direction, but that in endodermal lineages, there's only G2X, which produces a number of organs here. So that really clearly shows that there's non-random segregation. But this is in vitro and only for one chromosome. So we thought we're going to address this question and look how sister chromatid segregation behaves in our neocortical EMX1 positive lineage that are produced by radioglia cells. Now, we benefit from the MADM technology, and without going into detail, but the aficionados know that MADM relies on interchromosomal segregation here to reconstitute marker genes, GFP and tomato. That's why we have red and green cells and yellow cells. Now, what happens in a MADM event is that we're producing these recombinant chromosomes here, and depending how they segregate, it's against X segregation. So if they go to different cells, we get a red cell and a green cell. Or if they go together, we get the yellow cells. Now, this enables us to in a highly quantitative manner to actually distinguish X versus Z segregation. And to this extent, we define a so-called yellow, green, red index, or YAGRI. And this is really the number of yellow cells divided to the average of green and red cells. And now, if sister chromatid segregation is completely random, you expect a YAGRI of 1. In other words, it can be 50-50 chance to either go that route or that route. So in other words, 50X or 50Z. Now, when we look at projection neurons, cortical pyramidal cells, we were actually surprised that only for 2 chromosomes, chromosome 2 and 17, you get a YAGRI of about 1. So only here you get about 50-50 segregation, but for all the others, we actually see a biased segregation pattern that can be up to an order of magnitude higher here towards the Z segregation. In other words, we have way more yellow cells than red and green cells. Now you could argue, okay, this may have to do something with the efficiency of recombination maybe, but this seems not to be the case, because if we look at the chromosomes that show higher recombination frequency or the yellow cells and establish a ranking plot, there is no correlation of the non-random segregation bias with recombination efficiency. Now, lastly, we also looked in other cells, so cortical astrocytes in particular, and we see there the bias is much less. So it's only within a factor 2 or 3, actually, for the chromosomes that we are testing. But we also looked at CA1 pyramidal neurons. They are also positive for EMX1, actually, although different lineage, and cerebellar Purkinje cells. And we do not see any pattern. So in other words, for chromosome 7, we have a YAGRI of 2 for astrocytes, about 4 for pyramidal cells, hippocampus and Purkinje cells, and almost 9 or a bit more for cortical pyramidal cells. So we really believe there's chromosome specificity in how you segregate your sister chromatids, but also cell type specificity. This is summarized here. So basically, what I just said, for different chromosomes in a given cell type, there's a variable probability how you segregate your 2 sisters or sister chromatids, which can be 50-50, but there's also mostly a bias for most of the chromosomes into the other direction. And we looked at different neuron types, so also they differ from each other. And finally, we have also analyzed peripheral cells, T and B cells. They also show this phenomenon of non-random segregation, but it's also very different from the neurons here. So what we are proposing is that there is really a non-random sister chromatid segregation in the production of projection neurons, but also glia cells. And we're proposing that this could be a mechanism for asymmetric neurostem cell division. Again, the mechanism would be unclear, but there is evidence that chromatid-specific epigenetic modification, in other words, the green would be epigenetically modified differently than the blue, that this may correlate with the instructiveness of whether you're getting to a post-mitotic cell or a stem cell. So this is still work in progress. Part of it is actually put on a bioarchive if you're interested to read a bit more. Now, with this, I would like to thank the people involved here. So I mainly talked about the work of Nicole in the lab and also Florian in the first part with the PRC2 complex. Actually, Nicole is going on a job market right now, and also Robert Beatty is on a job market. So if you're interested in people who can master the MADM technology, these are two excellent candidates. I also want to highlight a talk by Andy Hansen on Thursday in this Neuromatch forum at 6 p.m. Central European time. And acknowledge a few collaborators, in particular Denis Jambordon, and also funding sources. And then I would like to thank again Guillermina and all of you for having me today.
So thank you, Simon, for this wonderful talk and this exciting data, and thank you for sharing this. So if you go to the question and answer button there at the bottom of your screen, you can read 3 questions that some of the audience have raised. And if you go fast, I think we will have time to answer the 3 of them if you can. Yeah. So can you actually read the questions so people, yeah?
Sure. So I'm reading a question from Maria Pompeiano. Sometimes you seem to compare somatosensory to visual cortex. Are the effects of EEG knockout the same in different neocortical areas? The short answer is we don't know. We haven't distinguished between functional areas, but it seems very important because what we do know is for the neurostem cell lineage progression and the clonal units that we saw, the number of 8 to 9 neurons from a radial glia cell produced, that seems to be invariant for different functional areas, but how the neurons distribute in the different layers seems not. So together with actually Oscar Marin, we found independently and in a collaboration that there seems to be region specificity of how you assemble the neurons. So it would be very interesting indeed to look at this. Yeah, definitely. Second question is by Pranyal Garg. Do you think it will be possible to induce neurostem cells in situ so as to generate neuroparts? How can we solve the issue of plastic variation of newly generated part and required levels of computation? Well, not sure if I understand completely the question, but I think this is very interesting. So there's a lot of data out there that imply that there is some remained plasticity, not only in stem cells as they pass in the lineage, but also for post-mitotic cells. So in particular, work from Magdalena Goetz shows that you can, and from many others like Benedict Berenger, you can actually reprogram post-mitotic cells to again produce neurons, in particular astrocytes. I mean, you can with a set of ectopic expression of genes, you can induce to produce neurons. So there is plasticity later. I'm not sure we understand completely all the mechanisms. We know that a few factors, a very small number of factors are sufficient actually. But also recently, Denis Jambordon showed that even stem cells remain plastic, in particular the radial glia cells, as time goes. So I think the underlying mechanisms of having that is unclear. Likely as epigenetic mechanisms.
Yeah.
Yeah. There's another question by Ella Mercer, and I'm trying to read. Hello. Oh yeah. Cortical astrocytes appear to have overall a lower YAGRI when compared to other cell types. Do you know why this is the case? Short answer, no, again, not. So this is very recent data. So basically the question is, the selective chromosome segregation bias is different for astrocytes than for projection neurons. Yes, we noticed that as well. We don't know. So what it could mean is that the level of bias is less important for astrocyte production. In other words, it could be other mechanisms that are much more important than selective chromosome segregation. Whereas for neurons, maybe it is. Again, we don't know the mechanisms, maybe epigenetic modification that could be involved here.
Yeah. I have a question for you if we have like 2 minutes. I see no other question from the audience. So there is a role that you have found in EED in the astrocyte, in the production of the astrocytes. Yes. Do you think that the effect is at a certain time point during development on the switch from the neurogenesis to gliogenesis on the progenitors that occurs something that makes the role of this EED in the production of the astrocytes, or do you think that is something set from the beginning of the progenitor's life that somehow epigenetically might be switched on a certain time point, like a clock that is, you know, switch on? I guess it's difficult to answer this. I mean, maybe you don't know, but it could be both scenarios, no? Or what do you think?
Yeah, in fact, actually, this was our preferred hypothesis that you just said. So in principle, when you look at Denis' paper, we did stainings, and we saw there's a sharp drop after embryonic day 12 in histone methylation. So in other words, the antibody doesn't recognize. But clearly, we see expression is still there. So there may be an ambient level of histone methylation still there. Mm-hmm. But what we think, what we thought is that maybe you have a priming mechanism that exactly sets the stage at the very early stage, but it's only translated later. Given we have also a cell autonomous function in the astrocytes later on, we believe that there's an active role for repression, and if you remove that, it's a separate function then for lineage progression. The problem is if we can separate the function of priming for lineage progression from the other function. At this moment, we believe it's relatively difficult because we also saw that the neurogenesis problem during the early lineage is really, if you knock out a gene in the entire neuroepithelium, not in single cells. So it may be that in the sparse knockout, the surrounding cells rescue these cells, and that's why they don't show a problem. But in the global knockout, there's so much deregulated gene expression if you remove that block that it may actually hide the priming concept that you were referring to. So it would be really nice.
Sure. Yeah, absolutely. Okay, I don't see any other questions so far. So thank you very much, Simon, as we have a very tight, slot. So thank you for your talk, and I hope you guys enjoy, everybody. So thank you for this. And see
Thank you very much, Guillermina. I really appreciate again the opportunity. I'm looking forward to all the other presentations, in fact.
Okay.
So, thank you.
Looking forward to seeing you soon. Yeah. Bye.
Bye. Thank you.